Welcome back to chapter 15, which is the lymphatic system. We're going to talk through how the lymphatic system functions, how it transports lymph, therefore lymphatics, through the lymphatics, the vessels within the lymphatic system, what are lymphatics, and the point of even having a lymphatic system, and then the immune response that is part of the lymphatic system. So first, this is a general picture of what's going on with the lymphatic system and what all is involved. So here we can start in the superior portion with the tonsils. We have three pairs of these, three bilateral pairs, and these are some specialized lymph nodes, and we'll talk about what lymph nodes are. Then we have lymphatic vessels. This is going to be carrying the lymph throughout the entire body, and eventually whenever the lymph is needing to be recycled, it will actually end up dumping into the right and left subclavian veins, which eventually dump into the right atrium of the heart. Then we also have the thymus. The thymus is primarily used at birth and through the first couple years of life, about till ages 9 or 10, and then after that it slowly deteriorates, but the thymus is responsible for making T cell or helping T cells become T cells. Then we have the spleen. Spleen is in the upper left quadrant of the abdominal cavity. It sits right behind the stomach. It's kind of sometimes hard to see. And the spleen is also part of the lymphatic system because, as you'll see, it'll recycle red blood cells and white blood cells. And then we'll have Peyer's patches. These are immune type cells that exist within the small intestine that will sample the environment at any given time and initiate an immune response if necessary. Small intestine, there you go. And then we have lymph nodes. We've got lymph nodes everywhere. You'll see a large majority are at the inguinal line, the axilla region, the cervical region, and you'll also see it in the popliteal region, which is the posterior aspect of the knee. And I think even in the abdomen here, we have quite a lot of lymph nodes right here as well. And so lymph nodes are kind of stopping points in this lymphatic vascular highway. All right, so functions of the lymphatic system. The whole point is to drain interstitial fluid from tissue spaces in order to prevent edema. So if you remember, our body is 70% water, right? And H2O. And this water has a lot of functions. It might be dissolving substances. It might be helping carrying substances. It might use its polarity to help move substances across membranes, for instance. It might also be there to help cool off the body. There's lots of different functions for water within the human body. But at some point, right, we don't want to accumulate too much water in one specific area. We need to have a method to move it from one space to another. And we need to have a consistent way of moving this fluid around. Don't forget, water is existing within the plasma of blood. It's also existing within the interstitial space, which is the space that is immediately surrounding blood vessels, nerves, organs, tissues. Basically, any space that is within the body will likely have interstitial fluid. And a large percentage of that fluid is made up of water. Where else is water? Water is also in cerebral spinal fluid within the head or within the brain and spinal cord. So we have water in so many places that we need to have a mechanism to move it around. If we have water or lymph not draining properly and being stuck kind of in one specific region, we'll have something called edema, which means swelling. And the swelling would be caused by those poor lymphatic vessels not being able to return the water or the lymph back to the heart. In addition, the lymphatic system is responsible for transporting digested fat to blood. So we'll see that in the small intestine, as we're digesting lipids, one of the last stopping points for lipids is a lacteal, and a lacteal is a specialized type of lymphatic vessel that will pick up lipids and transport them back into the lymphatic system and then eventually back into the bloodstream. We also have chyle, which is a mix of chyle, which is a mix of chyme and lymph type like lipids. And then that's what we call the lymph whenever it comes into the lacteal. Part of the other functions, as mentioned, is the immune system, which we'll mention briefly, and part of the immune system is to differentiate lymphocytes into B and T cells, which we'll talk about as well. So let's start with the lymphatic vessels. These are one-ended tubes. So they are open on one side and closed on the other side, and all of these tubes will have, there's big tubes, medium-sized tubes, and really small tubes. The really small tubes we call them lymph capillaries, and those are going to be sandwiched in between cells, because remember, the cytoplasm within the cells is also going to contain a lot of water and fluids. Eventually, all the lymphatic capillaries will join up with other lymphatic vessels, smaller vessels, up into medium-sized vessels, and eventually into larger vessels. And then finally, all of them will converge into two main pathways back into the heart for recycling. The main one that carries probably 75% of the lymph in the body is the thoracic duct, and then the other 25%. Maybe even less than 25% are carried via the right lymphatic duct. But both of these dump into their respective right or left-sided subclavian vein, or veins. So here's a picture of what's going on, the intricacies between an artery, a vein, and where the lymphatic vessels may be. So here we see a capillary bed of the arterial supply. And as things are diffusing back and forth and nutrients are moving back and forth, we'll also have lymphatics right nearby. And those ended tubes will collect, or those closed-ended tubes will have a higher pressure to to primarily push the fluid down into the bigger vessels of the lymphatic system and eventually back up into the heart. All right, lymph nodes. So all of these lymphatics will have stopping points along the way, and we call those lymph nodes or lymph nodules. And there's plenty of them throughout the body, as we mentioned in the previous picture. And entrance into and out of a lymph node, we have some terminology that we need to explain for that. So similar to the nervous system, we'll have efferents and afferents. E meaning exit, A meaning to arrive. Lymph will enter via afferents into the lymph node in multiple different areas. We might have multiple afferents, but at the end of the lymph node, after the lymph node is done doing what it needs to do, all of the lymph will exit via one efferent. We'll have trabeculae, which are we'll have trabeculae within each of the lymph nodes, and the trabeculae will be used to help not only separate and organize and structurally support different parts of the lymph node for where T and B cells might be. We call those centers. Oh, look, germinal centers, that's next. But they'll also be there to help structurally support moving parts within the lymph node. Those germinal centers in the middle or within the lymph node will produce lymphocytes. So there are some germinal centers that are specific to producing just B cells, or just storing B cells, and some that are just specific to storing T cells, since they have different functions. Here's a nice picture. The lymph node is actually a pretty difficult thing to really depict in a picture, because this image makes it look very organized, but it's not quite as organized as it's perceived in this image, but this gives you a pretty good general idea. So we have a bunch of afferents bringing in lymph into the lymph node. Then the lymph will circulate and eventually pass along next to these germinal centers. And in the germinal centers, we'll have B and T cells, and those B and T cells will help attack pathogens as they do. We'll also have some neutrophils surrounding that area. Neutrophils will clean up any kind of debris. They're not very specific. Same with macrophages. Macrophages will be eating them. And then we'll also have antibodies, which are produced by the B cells. And then any of the antigens that the antibodies are actually targeting might be in there as well. And here's a better picture of all of that going on. All right, so when the lymph is circulating through the lymphatic system, it will collect lymphatic fluid lymph from interstitial fluid. And the interstitial fluid, like I mentioned, is surrounding all the different types of tissue. Once it finally enters into the lymphatic system, then we finally transition the name from interstitial fluid into lymphatic fluid. Eventually, all of the lymphatics will form lymph trunks, and we have a trunk is usually where two or more things come together. Oh, and I don't think we're expecting you to know the names of the individual trunks. So, okay. How do we promote the circulation of the lymphatics within these specialized tubes? So, first of all, we'll have skeletal muscle contractions. So remember, there's smooth and skeletal muscle and cardiac muscle in the body. Skeletal muscle is voluntary contractions, and this is typically like our biceps, triceps, hamstrings, quads, gastrocnemius, soleus, rectus abdominis, things, those kinds of skeletal muscles. And as they contract and pump continuously, as you're walking, talking, breathing, sitting, moving your arm, whatever, all the vessels that are right next to it are going to be sandwiched right next to those blood vessels and the lymphatics. And as the skeletal muscles are pumping and pushing against each other, so if I were to draw like this, with the lymphatics or the blood vessels right down the middle, you can imagine as the skeletal muscle contracts, right, it's going to put a lot of pressure onto the vasculature in between them. And as it does this, it kind of like cinches off whatever is in the middle of it. And then it'll finally relax, and then the fluid can feel free to move in whatever direction it's supposed to move. So the actual contraction of the nearby skeletal muscle will help promote fluid movement. This is why it's so important when you're sitting in an airplane for a really long period of time, like 10 plus hours. It's really important that you get up and walk around because otherwise the fluid will just start to build up in your lower legs because you're not moving as much, you're not contracting as much, you don't have as much of the pressure to oppose gravity to move all that fluid back up into your heart. Same thing goes for just regular exercise. In addition, a similar type of mechanism is with the respiratory system. So as your lungs fill with air, and you can try this with me. So as you take a deep breath in, you can feel your thorax starts to enlarge, and your abdominal pressure, as you push out, as you push the air out, you can feel that pressure build up in your abdomen. So this exchanging of pressures between the thorax and the abdomen, that additional pressure is also putting pressure on the vessels, and therefore is also helping move lymphatics back into the heart, just similarly like we were talking about with the skeletal muscle. Then, of course, we finally have smooth muscle in the lymphatic vessels, similar to how we have smooth muscles in the arteries and in the arteries, in order to help contract and actually move things back up to the heart. Okay, so we do need you to know the trunks. Okay, so we have 5 different lymphatic trunks. The first is the lumbar trunk, which is going to be collecting lymphatic fluid from the lower extremities and the pelvis. The intestinal trunk, which is going to obtain lymphatic fluid from the abdominal region. The mediastinal and intercostal trunks are going to drain the thorax. Subclavian trunks are going to drain the upper extremities, and then the jugular trunk is going to drain the head and neck. Then all of those trunks will meet up with the 2 main lymphatic vessels that we really need you to know, and those are the thoracic duct and the right lymphatic duct. We have a nice picture here depicting what that slide was mentioning. So the thoracic duct essentially takes over 75%, or is responsible for 75% of the lymph from the left side of the body and the inferior portion of the body, while the right lymphatic duct is solely responsible for just the right arm, upper right portion of the thorax, right neck, and right head and face. So all of that dumps the right lymphatic duct, which you can see if you zoom in here, is going to dump straight into right subclavian vein, and then the thoracic duct eventually will dump into the left subclavian vein. All right, organs. Let's go into a little bit more details about what the organs are responsible for. So tonsils, like I said, we have 3 pairs of them. The names are palatine, pharyngeal, and lingual. Lingual under the tongue, palatine next to the palate, and pharyngeal in the pharyngeal spaces. These are responsible for protecting the nose and the oral cavity, which makes sense that we would need to have specialized lymph nodes in this area because these are immediately exposed to the external environment. Then we have the thymus, which we mentioned is helping to produce mature T cells. And I should put mature here. So T cells will originate from the red bone marrow, and in utero, they will travel to the thymus, and the thymus will say, hey, you're supposed to be a T cell, definitely become a T cell. And then it'll train it to be a really, really good T cell. And then only a certain percentage of those T cells will actually make it out and live the rest of their lives as T cells. As we mentioned previously, we have Peyer's patches. Those are going to be found in the walls of the small intestine, constantly sampling what's going on in the environment. Then we have the spleen. The spleen is going to be our largest lymphatic organ, our largest mass anyway, and its function is to break apart old red blood cells after the 120-day lifespan and platelets. And what would we need out of those that we can't already make on our own? Well, primarily hemoglobin and some iron. It will also be recycled as well. Iron is in the red blood cell. Spleen will also help produce lymphocytes and additional plasma cells as necessary. In case of a large hemorrhage, the spleen will also hold on to about a third of your reserve blood supply. So let's say, for example, you were in a Jaws movie and you were swimming out to sea because life is crazy that way, and your arm gets bitten off by a shark. You're going to lose a lot of blood very quickly. And so your spleen is going to kick in, recognize that you have a hemorrhage or some sort of shock to the system, and provide you with that extra reserve for a couple extra minutes, hoping that you can get picked up by someone else and then they can patch you up as necessary. So you can live without, you can kind of live without your spleen, but you're not going to live very long without your spleen. I forget the length of time you can live without your spleen, but typically if you have to remove your entire spleen, you're not going to make it, so maybe they'll remove just a portion of your spleen, a splenectomy. And one of the most common causes of actually having a splenectomy is car crashes, because the location of the spleen is just inferior to the rib cage, and it's right next to those floating ribs. And it's also just in your lower back region, which might not necessarily be the strongest or the most stable portion of your skeletal system, and it's most prone to actually getting damaged in a bad car accident. Right, here's a view of the spleen. Just like with most organs, we have a hilum, and a hilum is a area where multiple vessels are coming in and out from. So we have the spleen, or the hilum of the spleen here, and you can see the impressions of the other nearby organs on the spleen. Okay. The last function of the lymphatic system is the immune system, which is within the lymphatic system. And the purpose of the immune system is to protect the body from any types of or a majority of the types of pathogens, and also to help clear out debris from foreign substances that may have been existing within your body for X period of time, and now they finally have come to the surface, and the immune system has finally recognized them, and it's time to get rid of them. We have 3 different types of immunity. We have humoral immunity. Do we talk about the other ones? And cellular immunity. Okay, we don't talk about the other one. So humoral immunity, what does humoral mean? Humoral comes from the word humor. And that's not humor, haha, but that actually means like a body of fluid. So it used to be that anatomists used to think that the body consisted of 4 humors. One of those was water. And since they were right about the water, but not the other 3, anatomists decided to keep the term humor as part of defining anatomical structures. So anyway, we have humoral immunity, where we are actually having cells circulating in the fluid at any given time, and they are one of our best defenses in the immune system. So we have B cells, and B cells originate from the bone marrow, and in the bone marrow they are also turned into mature B cells, where they're told, yes, you are going to be a B cell. And those B cells, once they finally circulate and find invading pathogens, or they're told that they're invading pathogens nearby, then the B cell will respond by producing antibodies. And we'll talk about what antibodies are here in a second. But it will not only produce antibodies, but it will also produce additional B cells called memory B cells that will hang back and wait for the next time that they get exposed to the same pathogen, so that they are more quickly readily available to produce those antibodies again in case they get exposed to the same antigen another time. Then we have cellular immunity. This is with T lymphocytes. T lymphocytes, again, they originate in the bone marrow, but they travel to the thymus to become mature T cells. And these T cells are also traveling around in the bloodstream. They're just doing a little slightly different task than what the B cells would be doing. The T cells come in many different flavors. Many different flavors of T cells. Some of them go and tell B cells what to do. Some of them go and tell other T cells to regulate and protect the system. Some of those T cells go and actually attack pathogens directly by killing them off immediately. So that's why we have a long list here of fighting different various types of pathogens, because there's lots of different types of T cells that have very specific roles. So they get a little bit more praise than the B cells in some cases. All right, so what's an antibody? Antigens, we mentioned with the blood typing section. Antigens are tiny little proteins that exist within the body at any given time. The ones that are foreign are going to be recognized by antibodies. So antigens are recognized by antibodies. B lymphocytes are going to be producing the antibodies to respond to the antigens. And once they respond, or in order to respond to the antigen, they have to actually physically bind to the antigen, and we'll talk about where that happens. Eventually, antigens, or some antigens also have the capability of encapsulating a parasite or some sort of pathogen. So let's say if this was my little lonely amoeba floating around in my body for whatever reason, antigens might come and coat the surface of that amoeba and tell the immune system, hey, you need to come and destroy this. And then the immune system will come and later destroy this. So antigens have quite a lot of functionality as well. But in this course, we only really focus on their relationship to antibodies. So what is an antibody? An antibody has 4 separate chains, all made up of protein. And these are all genetically predetermined from your mother and father's DNA. And the way the 4 chains are linked, you have 2 heavy chains and 2 light chains. And the 2 heavy chains are linked by disulfide bonds. It's a Y shape, so it looks like this, and we have a picture of it coming up on the other slide. So these are our 2 heavy chains. They're connected by that disulfide bond, and then we have 2 light chains just next to the heavy chains. And then it forms this kind of Y shape, so I usually tend to shorthand it like this, which you'll see a lot of other instructors do as well. Then where do these antigens go? These antigens are going to be bound to the tops, which are our antigen binding regions. And antibodies are really interesting because they will have a lot of similarities across the board, but they will also be very unique to certain antigens at any given time. And that's really important because we want to be able to bind to any type of antigen that we might see or that we have previously seen. So here's a picture of the heavy and the light chains. And the reason why they're called heavy chains is because the heavy chains are similar across all, I should not have circled that part, I'm sorry. The heavy chains are similar across all species So your heavy chain is very similar to my heavy chain, that's what I'm trying to say. And even though they're genetically predetermined, they're going to be very, very similar in structure. And the heavy chain is usually what helps determine what type of antibody it is. And there's 5 different types. Then next to the heavy chain, you're going to have those 2 light chains. And the light chains are light because they're smaller. And they also help to determine what type of antibody it is. But they're primarily there to help build this variable region, which is depicted in green here. And the variable region is named the variable region because this is where the most variety comes from of the antibody. So the constant region of the antibody, let me do this in another color. The constant region of the antibody is going to be pretty similar across all of us. So you and I are probably going to have very similar constant regions, but we're going to have very different variable regions depending on what we've been exposed to, what type of vaccines we've had, et cetera, et cetera. However, your variable region is probably pretty similar to a sibling's variable region, but not as similar to your parents' region, your parents' variable region. And that's because as reproduction happens, hopefully the best genes get passed on, and those are the ones that tend to survive. So in theory, very slowly but surely, every generation will develop a better suited variable region that can better identify not only better identify antigens, but better identify a magnitude of different antigens. So that's why we care so much about antibodies. Antibodies are extremely, extremely important, and we really have to understand how they function. But at the end of the day, we wait for that antigen to come and bind to either end of the top part of the Y. Like I said, we have 5 different types of antibodies, come in 5 different flavors: IgG, IgA, IgM, IgD, IgE. And the Ig stands for immunoglobulin. Immunoglobulin protein. Like I said, antibodies are made up of protein. And then the letters G, A, M, D, E, I'm not sure if those were arbitrary or how they were decided. I'm sure there's some sort of reason out there. If you're struggling with remembering the 5, I believe the acronym is GAMED, which is almost in this order. Just got to flip these around. But let's talk a little bit about the functions of each one. So IgG is one of the most common antibodies, or the most abundant antibodies. Everyone's going to have some sort of mix of these 5, assuming that you're healthy. IgG is going to be one of the most abundant, as is IgD. So IgE is most abundant and IgD. So it makes sense that they're going to be having functions that are really, really important that we'll probably have to use on a daily basis, like attacking viruses, bacteria, toxins, and activating complement, which we'll get to here in a second. IgD is found on the surface of B lymphocytes, and actually whenever a B lymphocyte is maturing into a B lymphocyte, that's one of the major steps it has to pass before it actually becomes a B lymphocyte, is it has to produce an IgD antibody. Then we have the other 3. So I want to start with IgA. IgA is found in exocrine gland secretions, so basically mucousy areas. So think nose, reproductive, digestive tracts, respiratory tracts, things like that. So where it can live in mucus. And IgA is actually coming from your mother. So during while you're a very, very tiny baby and mom is breastfeeding, hopefully mom is able to breastfeed. And during that, or in the breast milk, many antibodies are able to pass on to you, and all of those antibodies are IgA antibodies. Otherwise, you can't really make IgA on your own, so this is the primary source of getting IgA. This is why it's so important to be able to breastfeed children in their first months of life. Okay, then we have IgM and IgE. IgM is the biggest antibody, the biggest in shape, and it will respond to bacteria or food antigens. It's big in that it has 10 different antigen binding sites, so it makes sense that it's going to be able to respond to bacteria, which are really variable and we're exposed to all the time, and food antigens, which we're also exposed to all the time. Then IgE, I always leave this kind of for the last because we don't usually ever go into too much detail about what IgE does. It's associated with allergic reactions, so places that histamine is released, and it's also really good at attacking parasites. But other than that, we don't usually talk about IgE too often. I said I was going to bring back complement back up. I don't think the slides talk about complement, so I'll go ahead and briefly mention it. Complement is another cascade system that's really important in helping recruit other white blood cells or enzymes to an area in order to attack a pathogen or a bacteria or a bad cell. So complement is just a name for a type of protein, but it's commonly referred to as complement cascade because you start with a complement protein and then that turns into another complement protein and then you recruit other types of complement proteins. So kind of similar to like the clotting cascade that we learned for the blood chapter, but specific to immunity. We have 2 different types of immunity once we have these activated antibodies and what they can actually do for us. We have active immunity and passive immunity, and each of those immunities have two separate flavors within them. So we'll have natural active immunity or artificially active immunity. Then we'll also have natural passive immunity and artificially passive immunity. So we have 4 overall types of immunity. And there are, right here, we have examples of the 4 different types. But let's kind of break this down one at a time. So what does active mean and what does passive mean? Well, active means that you have to actually activate your immune system in order for it to produce this type of immunity. Versus passive immunity means you probably got it from someone else. You didn't have to do anything. Either you went into the office and someone injected you with something, or maybe you got it directly from mom. But you didn't have to do anything. Like your immune system didn't have to respond necessarily to any sort of pathogen. Okay. Then we have natural and artificial. What's the difference? Okay, well, natural means that it's probably coming from another human or you're producing it yourself as a human. And then artificial means that it was either made by someone else or made by another species and then repurposed so that you can use it as a human. So now let's talk about some different examples between those definitions. So if we have naturally active immunity, this is kind of what we were talking about with all the previous slides with B and T cells. You get exposed to a pathogen, let's say the common cold, even though this is, let's not even do the common cold. Let's see. Let's do measles. So you get exposed to measles. Measles is floating around in your body. And the first time you get exposed to measles, you are going to activate an immune response. That immune response is going to take care of the, well, not really, but it'll take care of the measles. It'll get rid of the measles from the body. But it will also reserve some cells to remember to attack measles the next time that it sees it. So that's called naturally active immunity. Artificially active immunity is the same instance, except instead of actually being exposed to measles yourself, you'll actually have a vaccine which gives you a very, very small inactivated dose of the measles in order for your body to stimulate a response, but not a full-blown response, but enough of a response so that it builds those memory cells so that in case you ever do get exposed to the measles, that you already have an immune system built up to defend yourself against it. So this is why vaccines are so important. Passive immunity. Naturally passive immunity, this is, the classic example is usually maternal antibodies to fetus. So this is where mom is providing either breast milk to baby right after birth, or even in utero, passing on antibodies through the placenta. Or you have artificially passive immunity, like gamma globulins, which are usually for immunosuppressed folks. They'll have to go in and actually have this injected into them, or through an IV drip, I forget which. Also, another example is if you're climbing up in the mountains and you come across a poisonous snake, and you need the toxin to get rid of or you need to get the anti-serum in order to get rid of the toxin, you'll actually go into a clinic and get you'll get some sort of antitoxin, and that antitoxin is an example of artificially passive immunity. So, in summary, be familiar with what active immunity is and the different types, and passive immunity and the different types, but do not get tripped up by all of these words and how they all kind of sound the same and how you're using the same ones to define different things, okay? Take it one thing at a time, break it down, you know what the word active means, you know what the word passive means, you know what the word natural and artificial mean, and then put it in context for yourself so that you're able to explain it to someone else. Okay, let's go into detail a little bit about those B and those T cells. So B cells will originally respond to an antigen binding to its cell receptors, and as a result, it will make antibodies in response to whatever that antigen is. Then plasma cells will create, these B cells will turn into plasma cells, and those plasma cells will be making antibodies. In addition, those plasma cells will also turn into some memory B cells, and those memory B cells will hang around and wait for the next time that they get exposed to the same antigen so they can more quickly produce the right antibody. Then we have T cells. Like I said, we have different types of T cells, different flavors of T cells. And there are 2 main types of T cells, helper T cells and killer T cells. Killer T cells are also named CD4 cells, and helper, did I say that right? No, excuse me, I did that backwards. Killer T cells are CD8 cells, and helper T cells are also known as CD4 cells. So you might hear them both different ways, or both ways in the clinic. And in order to create killer T cells, you'll need helper T cells to initiate that process. That's not totally true, but for the purposes of this class, we can leave it as that. Helper T cells will also help stimulate B cells in order to make more of the appropriate type of cells. So helper T cells can tell B cells, hey, I saw this antigen, I think it's a problem, you should make this antibody. And then the B cell's like, cool, thanks, I'm going to make that antibody. And the T cell might also be like, hey, you might want to make this antibody. I'm not really sure if it's a pathogen yet. I'm not really sure if it's here, but you should probably make this antibody just in case. And so the B cells will respond that way too. CD8 cells, or killer T cells, or cytotoxic T cells, they're also called cytotoxic T cells. Cyto meaning cell, toxic meaning toxic. These will actually go and kill the pathogen, the infected cell with the pathogen immediately. It doesn't need anybody else's help. It can do it on its own immediately. Oh, perfect. Here we go. This is the killer T cells. So the killer T cells, like I said, are going to immediately attack those cells in one of three ways. These are examples of the different types of cells that a killer T cell might go and attack. Memory cells. We have memory cells of both T and B cells. So again, they're there in storage, ready to attack the same pathogen in case they get exposed to it again. We also have suppressor T cells. And suppressor T cells are there to help tell B and T cells, hey, slow down, we don't need your help anymore, like chill out, take a chill pill. All right, remember those macrophages we talked about a couple of times, and again in chapter 13. Remember, macro means large, phage means eating, so these are really big eaters. They will engulf and digest antigens and activate some sort of response, or at least help nearby white blood cells come and be recruited to the area to help fully get rid of whatever the pathogen is. We have lymphokines, monokines, and anything ending in the word kine is some sort of a chemical messenger, that can go and tell other cells what to do. So lymphokines means that they're going through the lymphatic system and they're going to be released by T cells, which is part of the lymphatic system. Monokines, means one, and those are usually, one cell. And these were one of the first cells to be discovered, and I think that's why the name is mono from the beginning. I'm not entirely sure on that, but anyway, monokines are going to be released from macrophages, which are also in the lymphatic system. We also need to talk about the skin and some enzymes that are important in the lymphatic system. So skin is one of our best contributors to the one of our best defenders for the lymphatic system, for the entire body in general, but one of the first barriers of protection for the human body. Not only is it a mechanical barrier, but we also produce sweat, or sebum, oily sweat is sebum, and those kind of environments, then the sweat being really salty, those kind of environments are really, really not pleasant for pathogens like viruses and bacteria. Viruses and bacteria like neutral temperatures, they don't like it too hot or too cold. They don't like really acidic or basic environments, and they definitely don't like being surrounded by other ions because those ions will flood into their cell and burst them straight open. So the skin actually is one of our best protectors. This is why it's so important to take good care of your skin and to stay hydrated and not to do any like laser treatments or bleaching treatments or shaving all the places. Just be gentle with yourself. Then we have lysozyme. Anything ending in zyme is a enzyme, and lyso is referring to lysosomes within the cells, and those organelles within the cells are able to produce lysozyme, and lysozyme is an enzyme that can help attack different types of bacteria. We also have mucous membranes. Mucous membranes are typically going to be directly exposed to the external environment, so mouth, nose, eyes, respiratory tract, reproductive tract, digestive tract, urinary tract. And it makes sense that we're going to have these membranes in those areas because those are going to trap bigger organisms and debris from actually entering into the body. And this is a very good defense mechanism. And this is also, again, why it's really important not to do things like enemas or to clean out your, what are those called? Like noddy pots? What what are those things called where you like dump water into your nose to help make it feel less congested. You actually are ruining and stripping that mucous membrane from those areas, and those mucous membranes are protective. Same thing goes for smoking. Smoking completely destroys the mucous membranes in your respiratory tract, making you very susceptible to obtaining a lot of organisms within your lungs. Then last but not least, we have hydrochloric acid, or HCl, and this is only in the stomach, and it's highly, highly acidic, pH of around 2, and it will immediately destroy microorganisms because those will just not be able to sustain themselves in that really acidic environment. All right, that's it for chapter 15. I'll see you again for chapter 14.